Hey guys, I hope you guys are doing all right. So before we start today's session, I want to go over a question from the Bacterial Genetic Study Guide. So this is that question. In E. coli, 4 HFR strains donate the following markers shown in the order donated. So essentially, this is the order that it came in to that recipient, um, the recipient strain. So these are the orders of all 4 strains. All these HFR strains are derived from the same F negative strain. What is the order of these markers on the circular chromosome of the original F positive strain? So what do? We start off looking at where these strains overlap, right? So in this case, the first one overlaps with the second one at WC, right? So WC, it goes this way, and then AL matches here, goes this way. And then we have BRU copied here, and then MZ comes back towards the 4, comes back to 1. This shows that the correct order of strains is this way. We do not include MZ at the end because it's already incorporated in the beginning. So that is how you would map genes depending on where it's located, right? And what time it actually comes in to the new strain. I hope that makes sense. Now we're going to move on to what is the central dogma of biology. I've seen this question asked in like every bio class I've ever taken, and a couple of my friends actually taught a couple of the upper level bio courses. So I've seen those classes actually ask this as a question. So forever remember it because it will come haunt you guys in future biology classes. So what is the central dogma of biology? The central dogma of biology is the saying where it goes from DNA to RNA to protein. So DNA is made into RNA through the process of transcription, and RNA is made into protein via translation. DNA is allowed to make DNA itself by the process of replication, and you can make DNA out of RNA through the process of reverse transcription. So just remember DNA to RNA to protein. Remember that forever, please. If I ever teach molecular bio class or something, I do not want any of you getting this wrong because I will just cry. Don't make me cry, okay? Please? Okay. So today's focus on the central dogma kind of leads into our focus for today. We're going to be focusing on replication and transcription and how RNA is processed after transcription, right? Okay, so to start off, let's do an overview of DNA replication. This occurs in the S phase of the cell cycle, and it is semi-conservative, right? We remember this from one of the very first lectures we've had in this course. So this just here is just showing the cell cycle itself. It is, as you remember, it is in interphase. There is 3 stages to interphase, G1, S, and G2. And the S phase is where in the cell cycle that it occurs. Okay? And synthesis always proceeds from 5' to 3'. So it adds on to the 3' end. Okay, so let's watch a video. Sorry about that. Nope. Come back here. Okay. Okay, so I like this video in that it kind of gives you an overview as to in explaining DNA replication as a process itself. So to continue on from here, it's more of focusing on what proteins are involved and what enzymes are involved and how they actually contribute into the process of DNA replication. So to start off with this, to continue on with DNA replication, it's just to focus down on these enzymes. So the first one would be helicase. Helicase is the one that unwinds and unzips and be like, hey, open up, we need to make we need to replicate stuff now. And to keep these two, so these strands, they love pairing back with each other. Like if they're close enough to pair right back with each other, these hydrogen bonds are like, nope, I don't want to stay apart. I don't want to stay apart. Let me go back to my pair, and kind of just goes back. So to prevent this from happening while replicating, they have these proteins called single-stranded DNA binding protein to keep them from pairing back with each other. So it kind of just stays away from each other. Then you have primase. Primase is required because you do not, so the polymerase itself cannot add on the different nucleotides without something to continue off of, right? So when this comes into play, we have an enzyme called primase. It adds on a primer, which is made of RNA. It can be different types, but in this case, it would be specifically an RNA primer that they would put in to continue off. So to continue off, they actually use DNA polymerase 3. DNA polymerase 3 extends it from the 3' end, and it keeps on growing from the 3' end of that strain, right? So remember, synthesis occurs from 5' to 3'. It adds on to the 3'. Then DNA polymerase comes in. Once everything is settled and whatnot, DNA polymerase 1 comes in and pretty much gets rid of the RNA primers and puts in whatever there is with and so the polymerase 1 actually removes that primer and replaces it with new nucleotides, okay? And the last one you have is to pretty much join those fragments together, especially in the lagging strand, because that strand is always discontinuous. So since it's different fragments going on, you need it to seal those fragments together, be like, yes, we're one happy family, we're gonna be all together, all sealed and tight, and nothing else can interfere with anything in between. Because if you have breaks in DNA, that leads to a whole lot of things that will be discussed in future classes, because that is so many things going on that will throw off DNA replication and how everything is processed. So those are the main enzymes involved in replication. And now we see where these steps are lying. So the first step is to open and stabilize the DNA strands. The second step is to prime or start off DNA synthesis with a primer. So you need a primase to put that primer in. Next is elongation or extension of the RNA primer using DNA polymerase 3 and these various nucleotide monomers, right? Without the monomers, we can't do anything. And then we remove those primers with DNA polymerase 1 exonuclease activity. Exonuclease is essentially just cutting out and degrading whatever nucleotide is there, okay? So after that, you join these. So those in the strands here, in the lagging strand, you end up with different fragments of these guys, right? The different fragments of DNA because it's not combined together. And the primer is like, oh, we need to make different primers every so often. So it ends up with different fragments, and ligase over here comes in and they join these Okazaki fragments together and just link them all together, and it's a happy new DNA molecule. So there is a lot of energy involved. And with this, you see that the energy actually comes from the deoxyribonucleotide triphosphate monomers. So essentially, each of the nucleotides, when they're added on, they release energy. So it's a lot of energy that's coming out of this thing. Okay. And DNA polymerase 3 is what actually participates in the elongation properties. It also has editing abilities to make sure everything is all set. Typically, it has a 3' to 5' exonuclease activity because it can go back and fix whatever it needs and then come back. Typically, mistakes happen 1 in like 10 to the 9th base pairs. That's like a lot going on over there. So it's not too much, thankfully, because if we had a DNA polymerase replicating and we had more mistakes per base pair, that won't be good, my friend. That will not be good. So thank you, DNA polymerase 3. So, okay. So the next part we're going to go into is a couple of questions regarding DNA DNA replication. This first question is, the bulk of DNA replication in E. coli is carried out by, what do you guys think? So the answer here is DNA polymerase 3, if you've guessed it. So next question here is, to replicate the single-stranded DNA at the molecule at right. So this guy here, this wonderful little circle. Which enzyme is required besides DNA polymerase 3? So what do we know here? We know that the DNA is single-stranded and we have DNA polymerase 3. What else do we need? So we can knock out helicase because that doesn't come into play. In this case, because it is already single-stranded, so you do not need helicase to open up anything. It cannot open anything in a single-stranded DNA. So that's out. Then we have ligase. Ligase only comes into play if you have so many fragments to come fix it up, right? So in this case, ligase is out. DNA polymerase 1 is typically to help out with more of the removing the primers, adding more nucleotides, that kind of thing. But since it's single-stranded, you just need DNA polymerase 3 for that factor. And it also has an exonuclease activities going on, so it can actually remove the primer if needed. So those three are out. So that means our answer is primase, right? Primase, you cannot just, the DNA polymerase 3 cannot just start adding nucleotides out of nowhere. So primase, we need primase to prime everything to initiation, right? We need to get things started. So primase and DNA polymerase 3 for a single-stranded molecule of DNA. So which enzyme in these guys are responsible for unzipping the double helix? So unzipping is essentially just pulling it apart. So what do we know? We know it's helicase, right? Yay! Which is the DNA strand that DNA polymerase constructs in the 5' to 3' direction. So which strand is this guy? Leading strand, right? If it's the opposite direction from 3' to 5', it's the lagging strand. Okay? Which enzyme can attach together the strands of DNA with double strand breaks? So what do you guys think? We're gluing things together, right? We're fixing up the nicks. So that means that would be DNA ligase, right? The discontinuous aspect of replication of DNA in vivo is caused by what? So what actually causes the discontinuous aspect here? It's because we have a restriction in the synthesis of synthesis, that it goes from 5' to 3', right? So it goes from adding on to the 3'. It is restricting the polarity there. So the answer to this guy is C. DNA polymerase adds new nucleotides to which end of a growing strand? As we've already talked about, it adds it to the 3' end. Okay? The enzyme required only for the replication of the ends of eukaryotic chromosomes is what? So we didn't really talk about this as much as I should have. Telomerase, so the answer to this guy is telomerase. Telomerase is an enzyme that extends the telomeres of the chromosomes. So these telomeres, in the process of like replication, it ends up getting cut short. So to make sure that you don't end up running out of these telomeres, these guys use the cells or in DNA replication processes, they use an enzyme called telomerase to make sure that the ends of the eukaryotic chromosomes are extended out. This is a typical problem in linear chromosomes. So that is why it is more specific to eukaryotic chromosomes, because prokaryotes are, as we know, they are circular. So they don't have that issue of ends getting cut off. Okay? Okay, so before I move on, that's pretty much what I have for replication. If you guys have any questions, please let me know. Okay. Now we're going to go into the topic of transcription. What is transcription? Transcription is the synthesis of an RNA, and it is the first step in trying to express a gene, right? So this is how a gene would be in transcriptional signal wise. So every gene has a promoter, it has a RNA coding sequence, and then there's a terminator region. So it stops it right there. So this process requires the different monomers, the ribonucleotide ones. So it has ATP, GTP, CTP, and UTP, which is uridine triphosphate. So adenine, guanine, cytosine, uracil. You have a DNA template and an RNA polymerase this time instead of a DNA polymerase, because polymerase, the word itself is an enzyme to add polymers, right? So to make polymers going on. In this case, we have RNA polymerase. That just means we are making a polymer of RNA, okay? There are key differences between eukaryotic and prokaryotic transcription. But before I get into that, I'm going to show you guys a video on transcription and how it occurs in a general sense. And then we'll go into more of the differences, okay? Okay, so one thing I would say about this part is that with introns, they do not code for anything, but we do not call them junk sequences because we do not actually know what they actually do. So it cannot exactly, they call it junk sequences. However, we prefer to just say non-coding regions because those, we don't know what these introns actually do yet. Okay, let's continue. Okay. So essentially what they were talking about, they kind of showed an overview as to what is happening in the transcription process itself. In so before we go more in depth as to the differences between prokaryotic and eukaryotic gene transcription, we need to understand how the structures itself are different. So this is a typical prokaryotic gene structure. We have a 5' end, it comes into a promoter, RNA region, coding region. This is where transcription starts. It goes and be like, oh, yay, yay, la, 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 la. And then ends up at this terminator region, which ends up as the name terminator. It stops everything, right? Okay. So anything before the promoter, it's called upstream. Anything after the termination site, that's downstream of a gene. Okay, it kind of comes in handy to understand these terminology when you go into like labs or different courses and they're talking, so downstream of this gene we have so and so genes, and we know that it is after that gene. Okay, so in a eukaryotic gene, we have similar structures, but some stuff are a little bit added. So we have enhancer control elements. So like we'll go more into this in later lectures when we talk about gene regulation and all that good stuff. But for now, you just need to know that there is a section where enhancers or suppressors, all those guys come in and control the situation that's happening here. And then you have a promoter region, which has a promoter that comes in, binds it to it, and then it starts off with so we have the part where it says exon, intron, exon, intron, exon, that is our RNA coding region. So when we do this, the DNA itself has everything there, the exons and introns. Exons are the actual coding regions. Introns are the non-coding sequences. As I said from the video, we just do not know what these introns actually do. So for now, they call them the junk sequences, but they are not junk. They wouldn't be there if we didn't need them. So let's move on to prokaryotic transcription. So the core polymerase is made up of 5 subunits: 2 alpha, 1 beta, beta prime, and this unit here. I forgot, it's a Greek letter. I just forgot what it was right now. So anyways, these are the 5 units that we have. We have 2 alphas, 1 beta, 1 beta prime, and this guy. I will eventually figure it out and tell you guys what it's called. Or if you guys have any ideas, let me know. anyways, we have those 5 subunits in the polymerase. So this is the polymerase just hanging out with all of the oh wait, I think that is sigma. I think. Yes. So we have 2 alphas, beta, beta prime, and sigma. Okay? Oh no, wait. Nope, not sigma. Something else. Because the core enzyme and the sigma factor, these two come together, they attach to each other and be like, oh yay, let's do this. Let's go find our recognition site. And they start, so once they recognize that at recognition site, they're like, oh, sigma factor goes bye-bye, and the polymerase actually ends up, the RNA polymerase ends up going, ooh, yay, transcribing, woo! And then at some point when it hits the terminator region, that's when it's like, I'm done. Bye. See ya. Sayonara. So that is essentially what happens with this guy. So this process is kind of row dependent. I don't know if Dr. Bruce goes this in depth with it, but essentially it comes in, it reaches that area, and it's like, bye-bye. See you later. So if a question comes in, draw a prokaryotic gene and its RNA product, be sure to include these specific components. How would you do this? So this is our DNA. Our DNA, we have our promoter, our region in between here, and then our terminator. So this is our coding area, right? So we have this area. Oh, wait, whoops. This, oh, okay. So this line is supposed to be here. Okay, so this is supposed to be shifted a little this way, and this line where transcription starts is supposed to be here. Oh, sorry about that. So when you look at this, make sure, I will change this in the PowerPoint, but this line here is supposed to be here, but it's meeting the promoter here, okay? I will fix this in the PowerPoint when I upload it. And then they do this, they kind of go in this way. They're like, okay, it stops here. So when it reaches the termination area, it stops. It kind of starts becoming the untranslated region of this side. So it kind of stops. It's like, I'm done. We're good. All is good in the world. Okay? So that's with prokaryotics. With eukaryotes, there's a whole lot of things going on. It's the same three steps: initiation, elongation, and termination. In initiation of eukaryotic transcription, you start off with a sequence, I guess a box to kind of bind to this TATA sequence. And so it's called a Tata box. The Tata box comes in, this process cannot start without the whole transcriptional activators. So these guys are The TAFs are transcription factors, activation factors. And this guy over here is the tactile box binding protein. So there are a couple of other transcription factors that actually bind to the tactile box. So this is the tactile box here. We have transcriptional factors that pull these guys in and allows it to activate so this is in the promoter region, right? So we have the promoter region. It comes in, binds here, and then we have this coming in. We have, so this is transcription factor 2D. 2B comes in and be like, with RNA polymerase 2 and 2F, like the transcription factor 2F, they all come in and it's like, okay, guys, we need to get this going. Woohoo. It kind of, so there are 3 types of RNA polymerases. In transcription purposes, we start off with RNA polymerase 2, okay? So with that, so once the RNA polymerase starts, it kind of continues on with like adding polymers and all that good stuff. And once it reaches that termination site, there is a specific region on the terminator site where it's like this region right here, AAU, AAA region, and it cleaves around 11 to 30 nucleotides away, and it kind of adds a poly A tail to it. So that kind of just shows how that kind of ends up happening. So with eukaryotic transcription, we have something that is called a pre-mRNA. With prokaryotes, this did not occur. But with eukaryotes, they have introns and exons and all of that going on in there. So they were like, okay, we need something to take out all of these non-coding, non-essential sections where you need, so you need these guys out in order to properly make the right protein. So in this case, we have this guy. So we have the pre-mRNA. The way that they would get it going is to actually process the pre-mRNA. And to process that, we call it a process called RNA processing. This processing method is actually only found in eukaryotes, because in prokaryotes, we do not have introns, right? So with this part, we know that these RNAs have to be modified. So how do we modify them? We modify them by a process called RNA splicing. So RNA splicing is essentially a way to get, one of the ways to splice an mRNA is to take out the intron regions, but sometimes it can take out exon regions too, depending on how it's being spliced, what is the control factors that are involved, so on and so forth. And there is something called alternative splicing. What is alternative splicing? It is just, so we have one way of splicing here. If you take these guys down, like, oh, you get rid of the yellow block here. But then here, when you do it this way, you're like red to yellow. Wait, no, my bad. So when you have red to yellow and then red to blue, you get this guy. When you do red, skip over yellow, go to green, and then blue, you get this guy. So that is kind of how you would splice these different mRNAs. And these different alternatively spliced mRNAs actually cause different protein isoforms. So these proteins end up with different isoforms, but we will talk more about how all of that would work with proteins in our translation session next time, okay? So a question here is, RNA polymerases add new nucleotides to which end of the growing RNA strand? Again, the same thing with DNA replication, it is from the 3' end. A particular mutation in the bacterial sigma factor allows this protein to bind to RNA polymerase, but prevents it from coming off the RNA polymerase once it binds. What effect will this mutation have on transcription? So in this question, it is asking what happens with the sigma factor itself. So it ends up preventing the formation of closed complexes between sigma factor, not the sigma factor, between the RNA polymerase and the promoter itself, right? Because when we know how the core polymerase and the sigma has to bind together to go meet the recognition site, right? So when that happens, it's like, oh, you cannot end up bind once the polymerase reaches the promoter, the sigma factor has to go away for anything to happen. So it actually ends up preventing that closed complex between the RNA polymerase and the promoter itself. I hope that makes sense. The bulk of RNA transcription in eukaryotes is carried out by which guy? So in this case, it would be RNA polymerase 2, because that is the version that goes for mRNA transcript, the mRNA, what's the word? Production. So RNA polymerase 1 is with the other ones, like, so all three of them have a specific role in which they create, okay? So that is all I have. I will have the study guide up as well, along with the PowerPoint. If you guys have any questions, please let me know. And thank you for watching. Till next time. Yay!